Great, thank you so much, and thanks to the organizers for inviting me. Today I'm going to talk about a little bit of some of the things that we've been talking about over the past week so far regarding looking at the network and looking at changes, and we're going to tie in a little bit of diffusion. But you can think about it in some ways that I'm going to take a much higher level view than we've been looking at today. And we're going to do this with regard to some sort of general biological questions, but almost exactly what we were just talking about. And that was, I might want to look at relationships in a particular species with one gene to another gene in a different species, or I might be trying to sort of look for changes. We might be actually trying to do exactly this where we were looking for changes in the organism. We might be trying to look at disease models. We might be trying to understand how the organism operates under stress. So an old school comparison, of course, would be looking at just strings. And as Ron said yesterday, we have much, much more information that maybe we shouldn't even be looking at this as a network, that we've got these sort of three-dimensional models. Here we've got one of my favorite genes, VEGF, one of Donna's too. And we look at this, of course, and we see that these proteins are actually 3D models. They physically interact. And there might be more subtle interactions too. We might be looking at a genetic interaction where it might be positive or negative, depending on it. And of course, we're going to look at this as if it's a network. So we are going to sort of take a slice of this. We're going to do an abstraction. So a high-level view of just genes as interactions. And I'll define for each of the particular applications what kind of interaction I'm talking about. But think dots and lines. So each of the nodes will be some protein, and each will be some kind of interaction. And what we want to address here is we want to look at this as a global scale on that particular graph. We want to see what has actually changed. So did I lose an interaction across some sort of evolution, or did I lose an interaction because of a stress? Or maybe I've gained an additional node, or maybe a whole entire piece of sort of the network is sort of drifting away in some sense. And what we've sort of done is we said, well, we actually know something more than that. We have reference points. And so with regard to this, we said, let's stop for a second and say, we have known nodes where we might know some information. Can we leverage those particular nodes that we have some information and in some sense seed, as it were, a matching between the graphs so that we could look for these individual changes? So here we might know something, say we've got the purple node, and we know that, for instance, this is something that's conserved. If I were doing something like an alignment, it might be a homolog. If I were doing something where I were interested in something around cancer, it might be a particular cancer gene that we know is in a few different types of cancer. And what we're going to do is we're going to sort of, just because diffusion seems to be the sort of topic or the underlying thing here, is we might actually pick our favorite distance metric. And we're going to see that we're not going to use only diffusion. And what we're going to do is we're going to sort of look at the graph. We're going to relabel it, if you will, with regard to these fixed sort of genes that we know some information about. And so what I could do here is, if you look on the two gray nodes, I could actually sort of create a vector, if you will, in the sort of graph space that would say I'm one away from the purple. So we let the purple be dimension 1. We let the, looking at this, we let the yellowish sort of be dimension 2, and our turquoise sort of dimension 3. And then we can label the nodes specifically to sort of recover, in some sense, where they are in the network. Now, of course, I can't, having been a theorist in my former life and being in this particular building, we might ask ourselves what it is about sort of graphs or networks to be able to sort of label them. Can we uniquely recover the entire network using a few sort of labelings? This has been done before. I'll just say this has been done before, and in kind of an interesting example, and that is the Pioneer plaque. So when we sent out this sort of into space, this particular Pioneer plaque, we wanted to tell everyone where we were. Whether this is good or bad or whatever your beliefs are here, they did so by sort of 14 pulsars. And the pulsars had a unique sort of signature for each one of these things. And what we did is we told everyone where the sun was with regard to these 14 pulsars, with the idea that people could find us. So this sort of idea of finding individual nodes from a mapping, from a reference point map, of course, has been around for quite some time. So the idea here, of course, is we want to create, if you like, a new representation. Lenore had sort of mentioned during the talk, we were looking at these diffusion metrics, and we said, well, we can think about this as an entire embedding, right? If I'm going to let a diffusion process walk completely over the graph, that in some sense what I'm looking at as an entire embedding of the graph with regard to some distance. What you can think about that we're doing is we're trying to reduce that dimension. We're going to try to actually look at this in a sort of smaller scale. So what we're going to do for each of these applications, we're going to select some landmarks. These landmarks, of course, hopefully have some kind of biological meaning, but we'll come back to sort of what they might be theoretical in a minute. And we're going to then label the graph whatever your favorite distance metric is. The first thing we should ask is this sort of in some sense feasible, being a former complexity theorist? And the answer is, of course, no. This is actually the metric dimension of a graph, so this is somewhat well studied inside of theory. I didn't know about it when we first came up with it, so shame on me, being a complexity theorist in my former life. And it turns out that the metric dimension of the graph is just looking and asking the question, if I were going to give you a subset of the vertices of the graph, how many would I need to be able to uniquely identify the rest of the graph? And here, of course, the distance is just the hop, just the next hop distance. And so if we look at this particular example, one of my little favorite little graphs, is we need 2 for this graph. If you look carefully here, we go through, we label it by the first one, it's at dimension 0 with 3 away from the other landmark, so on and so forth, and then of course 3, 0, 4, 1. If I try to do this with one node, it's not going to work. And in some sense, what's going to happen is there's a symmetry along the triangle here. But I don't want you to sort of be confused with just symmetries. It's not, this is a rigid graph, so there are no trivial, there's only one automorphism, the automorphism to itself. And so this isn't necessarily a distinguishing number. So people have looked at sort of the distinguishing number to break automorphisms. It's related. You would need actually the distinguishing number plus additional things. So it's strictly bigger than the distinguishing number. This is also inapproximable. So this means that there's no approximation scheme whatsoever. In fact, it's worse than that. So since we're in this building, it's actually W2 complete, which means it doesn't actually work even with the sort of factor. We can't do this as a sort of constant factor in any way, shape, or form. So no constant factor. It's not fixed parameter tractable. We can't really get at this. So you might say, is all lost? Well, again, we're trying to address this with biology, so we have information. We can just use what we have. We might hope that we're getting an approximation or some reasonable amount of the network, but we can still try. So for instance, we select the landmarks, we pick our distance, again, operated just for what we might be interested in. So it might be a diffusion process. We're going to label it, and then in some sense, this really is, as we've been talking about, an embedding into the metric space. So here, if you like, this, in some sense, this node B becomes an axis. And really, I'm measuring everyone's distance against that axis to that particular node. The I node is now another axis, and I'm measuring everyone against that distance. Now, this is not in any way, shape, or form going to be 2 dimensions. We're going to be working in things like 200 to 500. Now, the big claim here is that this is going to allow us to be able to make comparisons. So if I take one particular graph, say the graph on the top was embedded in this space, so it's here in all red nodes. I take a second graph here in the bottom with all blue nodes, and the idea is that I can somehow overlay the 2 particular embeddings in some reasonable way. Once we do that, we might be able to make comparisons across the networks in this new space. Now, I want to point out that making comparisons in this new space might be yet a different distance metric to be able to sort of cluster these particular things, and I'll talk more about that in a minute. I'm going to first do a quick application that's been very well studied here by many people in the room to the network alignment problem. So here, I might be working in a species, say mouse, and I want to make comparisons sort of on a global scale to the human network. And I want to look, in this case, I want to look for some similarities in some sense. What are the sort of things that have been preserved? There's a ton of work by many of the people that are in the room here throughout the entire time. So there's, and many more that I haven't even listed. Some new stuff that we heard about from Natasha earlier this week as well. So again, what are we solving with the network alignment? We're just trying to make matches, right? So I might have one gene, and I just want to point out these things might actually be mapping to 2. Just as we had sort of heard, we might have one particular gene that's either co-evolved, or I've got two genes that I want to map it to. Maybe I'm mapping 2 to 2. So there are lots of genes that shouldn't be matched. There's lots that should. Again, this is another one of these NP-complete problems because we're related to sort of subgraph isomorphism. So we're actually harder than just sort of graph isomorphism. I did hear in one theory paper that Babai said that the key to solving graph isomorphism was the metric dimension. The citation was personal communication, so unfortunately I don't have the paper. Some of this with alignment, the results were presented last year at ISMB. And the idea is we looked at this, we named it, as Ben says, you should always name it, that'll help things. So we named it sort of course alignment. Again, we're not looking necessarily at the same level where people have been looking at like the graphlet degree work where you might be trying to make comparisons. Here we're trying, on a local scale, we're trying to look on a much broader scale and then apply it perhaps to something that does, like Magna say, where you would actually go through and optimize for local conditions. We're trying to look for just functional matches. We're not going to use sequence. So there's something that I want to sort of say right out. We're not going to actually use sequence in any way, shape, or form. Many of the previous work, particularly isorank and Hubble-Align, use a lot of sequence to influence how matches are made. We're going to use structure alone. Now you might argue, wait, Ben, you're using a little bit of this because you're going to use some homologs, and that's true. We're going to sort of seed it using a little bit of sequence and then ignore sequence from there on out. And so just as I alluded to, when we do this, we're going to actually do alignment. Biology told us something with regard to who should be the special nodes. Here, we're going to use homologs. And I'm hoping Roded will ask the exact same question that he asked earlier, which is we did the same thing. We're going to throw out any homologs that actually are multiple maps. We're going to sort of avoid them completely. So we are going to do one-to-one mappings only. We take them from homologene, and we're just going to sort of say that's our sort of seeded network for this particular problem. In our preliminary results, we actually used DSD again, that Lenore had talked about, where we're using a diffusion distance where we're going to downplay hubs. And so to here again, just a reminder for everyone that saw it, you can think about it as, I always say, who do you want to set up in a date? Do you want to set up A and B, or do you want to set up C and D? And the idea is most likely A and B, because there's a lot of interactions of low degree as opposed to sort of one that's sitting in the center. I do want to also say that we're looking at this. It's not required that we use DSD. In fact, actually, we've been studying the human network, and we're realizing that some of the diffusion techniques that's from the Raphael lab are actually a little bit better or seem to correspond closer to things like Resnick scores. So here we're looking at maximum information content using Go. And so using a sort of labeling somehow, it does seem to be that some of the heat things work better on the human network. DSD outperforms on the yeast network. I point this out because this technique does not require that you use the same metric on the same network. You could pick your favorite network or your favorite functional sort of measurement on yeast, then pick your favorite functional measurement on human, and then actually embed them into the space and make a comparison across the space. So again, we're going to sort of take the two networks, we're going to align them, and ideally what we'd want to do is sort of find a nice clustering metric. So we'd want to sort of find a way to sort of cluster these. Again, for preliminary results, I'm going to sort of force things to make one-to-one matches. So for now, think I could cluster. Right now, just to make it easier, I'm going to do a sort of one-to-one matching and sort of the best. And so again, how do we do that? We're doing a similarity across species, and this picture is a little bit of a misnomer. And part of, if you think about it carefully, I might have the yeast network and I've got maybe 500 landmarks, and I compare it with 500 human landmarks, but the network sizes are so different that actually the scale on this is very different. So when I say I'm going to align these and suddenly I'm going to get blue dots near red dots, it's a little bit of a misnomer with this picture. What we're really going to do is we're going to take all of them in one network, all of the other network. Now remember, each one of the dots is in like a 500-dimensional space. So then I'm going to take something to be able to sort of compare or reduce that yet again. And what we did is we either do L1 norm if you're about the same size. So if we're comparing the same network to a different network under stress, we might do something like L1 norm. If you're going to take the yeast network or the mouse network and compare it to humans, you would do something like a cosine similarity. Am I sort of at least in the right direction in this multi-dimensional space? So think cosine similarity for any time I'm using species that are very, very far apart. Think L1 norm if I'm doing something where I know the number of nodes is exactly the same. Good? And again, these are fake numbers here, but what that's going to do is it's going to give me an all against all matrix in some sense, a sort of similarity matrix for each one of these particular genes. And then I could do something like a Hungarian algorithm or something more sophisticated like cluster. So again, preliminary results. The PPI network, so unfortunately Fly is sitting right there in the middle. Natasha, I apologize. I had no idea. But apparently now I have a new yeast network that I can't wait to try out too, so that's pretty exciting. We use string, a little bit older version of this. Just to give you some ideas, the human network is about 14,000. The yeast there is going to be 5,000. Now, anybody that's a gene counter is going to realize that these are a little bit lower numbers. That's because we take the largest connected component. And in fact, you should probably actually take the 2-core as well to get better results with regard to diffusion. The 2-core is, of course, going to remove all of those sort of dangling tails. Donna sort of mentioned this in her talk. If you've got a dangling tail, you can't make a comparison to another one. That's an automorphism automatically, right? So all of those. The other thing you might think of is disconnected components are always going to connect to other disconnected components. So the reason we don't do it is DSD, these diffusion techniques can't be computed is the real reason. But you can think that I can't break those automorphisms as well. We're going to use Go in terms of our functional comparisons. To test it, we wanted to know, does this in some sense, again, these diffusion techniques, do they sort of work with perturbed networks? So could I start with a ground truth? And so we created tests with ground truth. And the way that we did this is we took the mouse network. We picked the mouse network because we wanted to pick one that's sort of big in some sense. We took it, we cloned it, and then we perturbed a copy. And we did this multiple times. We chose the landmarks at random. Because I'm using the same particular network, I could choose the sort of homologs or preserve things. We actually ran tests on degree, whether you were high betweenness scores, high degree. Random was best, to be perfectly honest. I hypothesized that this is because randomly is in some sense getting a better closer approximation to this actual metric dimension of the graph. And you can think about it as it's just going to be spread out. They're going to be sort of spread out in such a way that we'll do better in some ways. We use the Hungarian just to be able to create a perfect matching, and then we go through and take a look at how it did. Again, thin, what we did a perturbation where we thin the network. So here, we're just going to sort of assign probabilities to every edge in the graph, and we're going to move everyone underneath that threshold. So we could sort of do different levels of sort of thin across this. Similarly, we did a rewire. Here, we're going to sort of pick it up and move it at random. So if you're below the threshold, I'm going to sort of pick an end at random, and I'm going to sort of force you to join someone new. And we force you to join somebody new. You don't necessarily sort of land back on yourself.
Where did you get the probabilities?
Assign them randomly. So I'm just going to go ahead and assign it randomly across the edges. No, and it's just there's no interesting distribution at all. And then Scramble. We picked it up and we sort of completely said you've got to go somewhere else. So we moved the edge. Again, not a rewire, but you said you've got to go find somewhere else to actually do this. And so how did we do? We ran it all the way to the end. Just to sort of see how it is. And you can see pretty much we drop off pretty quickly, as does everyone else. I want to point out to a lot of the other sort of competing ones, they're meant to be done with sequence or with additional information. This is structure only. So these are not necessarily the way that you would run these other competitors. We're looking at just structure on itself. And so what we see with using diffusion is, in fact, you do get a sort of break or a slow sort of curve here. And one of the reasons I think that's possible, again, is because diffusion is sort of more robust in some sense to noise. So we see for each of the sort of tests that we're sort of, in some ways, sort of tailing off a little bit slower. I do want to point out, since this shows up in reviewers all the time, that you do care about this huge amounts of perturbation. You might say, why would you destroy a network to 50%? Because you must be at noise. I will, of course, remind everyone there are 5,000 inside of mice. There are 15,000 inside of humans. The networks are more than 50% different. So we do need to find a sort of robust technique to sort of find at least the coarse alignment, and then maybe do a finer granularity. So it's one of my personal beliefs. What happens with no ground truth? So here we looked at this and we said we want to actually do this on real networks. So we tested it against all of the networks. We're going to do a measure of performance here with biological similarity. There are obviously structural similarities that many of the people have come up with in the room, S3 and ICS, for instance. We looked at biological and functional similarity, partly because we don't make good matches at a local level. So we will not actually get good scores for ICS or S3. What we get is something that's much more functional. So again, homologs are my landmarks. We did 3 types of validation techniques. We did the functional consistency. Here we're looking at, do I match a particular Go term? We did the Jacquard Go. Here we're going to actually look across and sort of see the number in some sense, or the amount of sort of annotation that matches. And then we did a kind of cross-validation. And again, this time we did a Hungarian algorithm, although you could use and probably should use something more sophisticated. And we tested this against, and we do pretty well. So we compared to, again, Hub-Align and Magna and these, we're doing just as well, sometimes a little bit better on human and mouse and yeast to human. But this is a shared Go term. So I do want to say this means that there exists a Go term. This Go term could be biological process. So in some sense, you know, saying I've got 70% match, this could be at a very, very high level. So Jacquard Go is a much sort of better way to sort of think about this. And then you can see Hub-Align actually does quite well. Hub-Align uses a lot of sequence again, so there is going to be sort of a bias here. And then we're on standard for these things, maybe a little bit better on each of them. One of the things I think, though, that's more interesting is we take now and try to do a cross-validation on homologs. And here what I do is I take 50% of my landmarks, and I make that the target set. The other 50% are my anchors. So I take all of the sort of homologs that I have, and I hide half of them. And we just ask, can you recover them? To what degree can you actually recover them? Well, now, again, this isn't at cluster level. These are the perfect matches. With the perfect match, do I get it back? So it's pretty strict. And we see we do quite well on, actually, unfortunately, some of the fly. But we do well on sort of worm and yeast. Here, where these things are a little bit less, IsoRank actually does well on mouse. And Hub-Align does very well on the mouse to human. So we're doing quite well when we ask for these exact matches. And again, this is the Hungarian, so we might be able to recover this when we do something more sophisticated at a group level. Then we actually sort of said, well, we don't use sequence. So we didn't test to sort of validate with sequence, but what we said is, do we actually get to find sort of a novel match in some sense where I might make a functional match? Because if this is truly a functional embedding, can I make a functional match? And so what we did is we sort of just calculated the Resnick score. The Resnick score, again, is the maximum information content. So here I'm going to actually look at the annotation across the corpus. I'm going to look at how detailed, in some sense, is a measurement of how detailed your annotation is. Not do we match on biological process, do we match on something that's very specific. And we just ranked them. We just found our sort of top 10 in some sense. And what we found is we found homologs, so the sort of top 3 in terms of our score. Were in fact homologs that weren't part of my system, things that I didn't know about. So these aren't landmarks, these are just ones that I didn't do. They maybe were the ones I threw out because they might have a multiple match. But then what we looked at is we had high functional scores and low blast bit scores with long sequence lengths. So here we've got these things that aren't necessarily based on sequence, but do seem to be related in function. And then we went back and sort of said, all right, let's actually look all of these up. And we saw things where we had similar arrangements, similar structure, coordinators, regulators, things that have been known to be biological or have a biological citation that says these things look like they're the same. So we were quite pleased with that. Now, we did this with the idea that we wanted to make comparisons. But we also said, well, wait, can we actually look at this and try to do differences? Now here, I might be the exact same network, the exact same number of nodes, but I now want to look at the amount of rewiring that's been going on. Again, at this sort of global scale in the network. So here, it's a little bit different. We calculate the same score. We'll use an L1 norm because I actually have exactly the same scale, so L1 will work just as well as sort of L2 or even a cosine similarity. We actually then only care about the diagonal. Because the idea here is my network is exactly the same. My node matching is known. It's the same network. I know what the node is before. I want to know, has it sort of moved or has its pattern sort of shifted in the network? So now I care only about the diagonal. And again, I'm looking at how much has it changed against the target set. This is very preliminary, as in the last couple of weeks. We looked at this in 2 different sort of things. The first one was we looked at genetic interactions. The Krogan lab had 2, the original sort of, I guess, wild type of the genetic interactions, and now the MMS damaged. And they looked at this, and they said, well, what are the rewire things that have sort of happened when I do sort of DNA damage? And what we looked at is 2 different types of landmarks, high degree and static nodes. We use positive and negative, so as Anastasia was telling us before, we should separate those things out to get a better signal. Right now we wanted a little bit bigger network, so we dumped them all in together. We use DSD as a distance, and what we found when we sort of looked at the sort of top things that percolated all the way to the top, the most sort of change in the network, were in fact reported as MMS repair. The highest in both, static in this, was actually a transport gene. And the idea, so this was actually an undergraduate that did this work, and she ran to her biologist professor and said, why is this happening? And she correctly, or sort of hypothesized that, of course, the transport is going to clean out the cell. So if you damage it with DNA, you're going to see this sort of signal from a transport because of sort of repairing or cleaning the cell. This also actually was great that this undergrad ran and did this, because what she did is she found a gene that her professor was interested in that had already been looking at this with regard to MMS. So this is now in the wet lab this past year to sort of test, is it in fact sort of something that we should be caring about with this MMS repair. So we're a little bit lucky just because the undergrad went and asked questions. And another sort of network, again, looking at the sort of differential expression that we've been thinking about, and this is pretty preliminary work as well, is a co-expression network. Here we're looking at breast cancer. We're looking at sort of the estrogen receptor, sort of whether you're upregulated or downregulated. This is work of one of my seniors that's graduating this year. And what he did is he sort of collected expression networks from a couple different experiments, and he sort of used the standard technique to sort of transform it into a network. What we did is we actually sort of, rather than cutting off that network, we let everyone sort of vote on their edge. We let that sort of probability or confidence of that edge, we gave it a weight. We take the negative log to sort of, in some sense, translate it to a distance. The landmarks we used were upregulated and downregulated. This was just from the Broad. And then since this distance is a weighted network, we actually used shortest path. So we looked at sort of shortest paths across this. And then we did a cross-validation test. And over 1,000 runs, we hide half of these sort of 480 implicated genes. And the other half are sort of way far upregulated, or much higher sort of scores than we'd expect. And then we did random landmarks for 30% of the genes, just to sort of, in some sense, 480 is not very big. We think the metric dimension of humans is probably around 800, actually, from our test on that network. It looks like about 800 is about our metric dimension. So we took 30% of them, and we saw that the up and down regulated were consistently higher. And this time we used the cosine similarity for that one. And with that, I'm just going to sort of go through and thank all of my co-authors. This is actually work of tons of undergrads. My collaborator, Mark Crabella at BU, and the rest are undergrads. Inbar Fried is just graduated. He's at Harvard Medical. Tony Canistra, who's also worked with Ben Raphael, graduated last year, and he's going to start at the University of Washington in September. Jennifer Hamelman, who did a lot of the gene interaction networks, is leaving me for MIT next year. Thomas is going to join Ben at Brown in the fall, and Taher is actually going to Johns Hopkins. He's also a senior. So these are the results of many different senior theses. So with that, thank you very much.
So about cosine.
Yeah.
So, to relate the question, so if the networks are relatively the same size, does it matter whether you use the norm or cosine?
It's not so much. We did notice that the cosine seemed to be performing a little bit better on the ER plus and ER minus networks. So we were using that. So we've been defaulting to sort of cosine in some sense. But I don't think that there's a strong difference between them, to be perfectly honest. I think there's more work to do, though. And the way that we should think about this is, let's say that you and I are sort of close to a particular homologue. That's really interesting. But both of us being far away from a homologue, that's not as much information, right? So my belief is that probably we could find a bit vector, sort of somehow use a bit vector carefully in some sense, and then actually get better than both of them. Like if
I guess you almost answered my other questions.
Yeah, please.
About the philosophy about cosine. As you know, you have smaller network, you have bigger network, and somehow, I think underlying philosophy under cosines that the bigger network will be somehow uniformly bigger.
Yes, and this is absolutely not true. Yes, yes. So I think actually what we should do is a much more sort of careful. In some sense, you could actually imagine weighting it, right? So these landmarks, perhaps maybe if you were doing something that you were interested in, you'd weight a landmark more. So the fact that we were sort of close on a particular two homologs might sort of blow out the whole entire score and say, okay, that's it, they should be in the same cluster. So I can see a situation where you might weight the landmarks differently because of what you would be interested in, and far doesn't mean anything. Both of us being far mean there doesn't seem to be sort of anything interesting with that. So I think cosine is our sort of first attempt to try to get at this sort of scale that we tried L1 and it didn't work at all, right, on the human network, because you're blown away with these big differences that mean absolutely nothing. So we at least said, okay, cosine might get us in the angle, but as you say, they're not uniformly different, right? They're morphed together. So I think a bit vector technique of some sort that we carefully apply would be best.
Well, oh yeah.
Oh, please.
So it seems like you want to have as many landmarks as
You want,
possible.
yes, yes.
And then if you have two networks, if you have a gene that's disconnected in one but connected in the other,
It's
then
gone.
It's gone.
It's gone. All games are off. This is an artifact by using the diffusion pieces because there's no way for... so essentially all of the diffusion techniques that I do... so one thing that's great is, well, it's not fast, but the technique to calculate this is n cubed. So it is a couple hours in some sense as opposed to sort of much, much longer time. So I can calculate and do this sort of in a couple hours, even for sort of humans. And so, but what I can't do is I can't do anything with a disconnected network, just because there's no inverse for the graph. Yes, please.
Your simulation experiments, you showed how the performance drops with the
Yeah.
error.
Yeah.
That, if you put that aside with the actual errors in the real known networks that we know of, how do you reconcile these two?
So in terms of the ones, that's actually a very good question. So the question is in some sense you see as we perturb this off, these networks are actually more than 50% different. So how does that sort of relate in some ways? And I think again, what you could do is something like, and I'm going to look right, you could do something interesting like take Magna, where Magna will optimize on particularly what you're interested in. You would take our sort of technique and say you want sort of a coarse alignment in some ways. We'll get sort of just down to the region level where these might be functionally interesting, but then you get locally to try to sort of make a better alignment. What I would actually argue is, in some ways, we want to sort of get at a sort of coarse sweep first because we're doing something global, and then do something interesting sort of that is even better studied. Yes, please.
I've never seen such a great coverage of existing work on network alignment.
So fast. Yeah, in 20 seconds.
Questions and comments. I'm very passionate about the topics. So my question/comment, just want to hear your opinion. Is it really fair to compare raw scores of these different methods, given that they are all producing different output types, such as one-to-one,
Absolutely not.
multiple, many-to-many?
Yep, absolutely. So.
Compare p-values of their scores.
Absolutely. What we did for the structural part, so looking at the sort of perturbation test, right? And as I said quickly, like this isn't a fair comparison because what I did is I stripped away everything, right? So other techniques are going to sort of find and make more sophisticated matches. I forced everyone to do Hungarian. And then I used just that sort of matrix for the idea of sort of saying what similarity matrix are we looking at. But I make no comments about sort of, that it works better than Magna or at all. Because we're not trying to, in some sense, solve the same problem. In fact, I look at sort of Candle as sort of a precursor, right? So where you might actually then sort of use something that would optimize around a sort of local piece. So it's not a fair comparison at all.
So I think
No. We should work together.
okay. Thank you.
Thank you. Thank you.